Good day, this is Professor Will, CBMD PhD, and today is Wednesday, June 16, 2021. It is 6:15 and 35 seconds a.m. Eastern Standard Time. Today's research, cancer cells, and don't forget to check out my website. Well, let's hop to it here. Cancer cells fight for their footing by using an aging gene. Date June 15, 2021. Source, University of Helsinki. New results allow the development of novel therapies for hereditary forms of intestinal cancer. Researchers have discovered how mutated cells promote their chances to form cancer. Typically, the accumulation of harmful cells is prevented by active competition between multiple stem cells in intestinal glands, called crypts. The functioning of intestinal stem cells relies on growth factors, named WNTS, produced by the surrounding environment. Intestinal cancers typically originate from stem cells where mutations allow growth independent of these factors. When researchers removed a gene called NOTAM, which renders WNTS inactive, from mutated stem cells, the number of precancerous adenomas in the intestine was greatly reduced. Researchers found that mutated cells use this gene to block environmental factors critical to normal stem cells, gaining advantage in competition. Researchers previously discovered that the same gene, also called the aging gene, is expressed in normal tissue when we age, reducing the ability of stem cells to repair damage. The current study shows that mutated cells use the same gene in order to establish a permanent footing in the tissue. Mutated cells kind of hijack the aging gene and use it against the healthy stem cells. The results from this study may lead to the development of new therapies because the function of the enzyme encoded by the aging gene can be blocked pharmacologically. Researchers previously used a compound for this purpose in aged research animals to enhance the function of aged stem cells. In the current study, researchers used the same method to reduce the chance of mutated cells winning in competition. A 3-week treatment reduced the number of adenomas in animal models. The results are promising and create a foundation for developing therapies for patients predisposed to intestinal cancers. This research demonstrates that by enhancing the natural mechanisms of how tissues remove damaged cells, we can also reduce cancer risk in other tissues. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.